Hello, scholars. We are in lesson 4 of unit 6, genetics. And in lesson 4, we get a little bit more detail on the process of meiosis. Now, I know we talked about this briefly in Unit 5, and I'm just going to hop over to a slide in Unit 5 I want to use for review here. But in Unit 5, we talked about mitosis primarily, and mitosis is when cells go through the cell cycle to make identical diploid copies of a cell for growth and repair. You bit the inside of your cheek. You worked out really hard and your muscles are growing. You're a baby growing into a bigger human. Those are all mitosis events that take place through the cell cycle continuously in our body. And the I in mitosis should make you think of identical if we were doing a crossword puzzle. In meiosis, the E is for sex because in meiosis, you are producing sperm or eggs. These also have sometimes these are called gametes or sex cells. So there's lots of instances in biology where you have multiple terms for the same word. So, meiosis is making sex cells, and mitosis is making identical somatic body cells. We talked in Unit 5 about the differences between how these types of processes are passed on. If an injury or something occurs during mitosis, that remains with you. You're not going to pass that on to your offspring. You're not going to lift a lot and then pass big muscles onto your offspring. In meiosis, if errors occur or if there are mutations that are passed into your gametes or your sex cells, they do have the potential to be passed on to future generations. So, those are the big differences there. And I just wanted to bring that back to your attention. A closer look at mitosis. Homologous chromosomes with the same genes, one originally from each of your parents, are diploid, containing both sets of chromosomes. And this is also sometimes called 2N. 2N means you have one set of chromosomes from mom and one set of chromosomes from dad. 1 plus 1 equals 2. In a haploid chromosome, you're going to have only a single set of chromosomes, and that would be 1N. So this would be a 23 chromosome cell if this was a human, and this would be a 46 chromosome cell if that was um, if it was a diploid cell for a human. Diploid cell for a human, 46. Haploid cell for a human, 23. I apologize. All right, so just reviewing mitosis, remember you've got prophase, metaphase, anaphase, and telophase. If you do need to refresh your memory about any of this stuff, you can always go back to the Unit 5 slideshow where we talked about that in detail. Meiosis is 2 stages. You have meiosis 1 and meiosis 2. They go through prophase, metaphase, anaphase, and telophase, and then they go through it again: prophase 2, metaphase 2, anaphase 2, telophase 2. And the big takeaway you should see here is instead of having 2 genetically identical cells at the end of the process, you end up with 4 genetically different haploid cells at the end of the process. What's really important to think about, not just for this unit, but we will be building on this moving forward, is that during prophase 1 of meiosis, something special occurs called crossing over. And what happens is these chromosomes kind of swap information with each other. So at the end of prophase 1, instead of having a red chromosome and a yellow chromosome, now you have a red chromosome with a little bit of yellow on it and a yellow chromosome with a little bit of red on it. And as we've said before, this is going to increase genetic diversity, and it's also going to help ensure that you're not identical clones to your siblings. The genetic information gets scrambled a little bit in here, so that way we have some genetic diversity. I just talked about that. Here's another picture of recombination, another slide, because it does show that it does generate genetic diversity, and it is really important. And on this image, you can see how the letters and colors are crossing over to create chromosomes that have just a little bit different gene combination. Another picture of crossing over. They really crossed over in this one. All right. So, genetic diversity is helpful when we examine sexually reproducing populations. And we'll talk about this in Unit 8, but it helps those populations to survive in their changing environments. In other words, genetically diverse populations are more likely to survive changing environments because it makes a population better suited to adapt to changes in their environment. We're going to talk about the leopards a little bit now. We'll revisit them in Unit 8, but leopard populations around the world are in danger because of inbreeding. Their range is much smaller than it used to be. So, if you were this leopard and you were used to walking around and grazing and traveling very far and going into a different leopard population and looking for a mate, now you might not have as much option. You might not have as many choices for mates because your territory, your habitat has been reduced and there's not as many individuals that you can pick to mate from. So, unfortunately, they end up mating with someone who's maybe a little too closely related to them, and they have very little genetic variation between any of the two individuals. This means that if there are any kind of genetic disorders that are passed on from parents to offspring, you're going to see more of them in the population. It also, low genetic diversity also makes organisms very susceptible to disease. And it is predicted that at some point, probably not too far in the future, this is going to lead to their extinction. And this is a problem that many large mammal populations are facing due to habitat destruction, due to human interference. They are becoming less and less genetically diverse, more susceptible to disease, less able to adapt to a changing environment, more likely to become extinct. So we need to think about genetic diversity a lot. Recombination that occurs because of meiosis through sexual reproduction and mutation, which occurs randomly, provide for genetic diversity. Inserting, deleting, or substituting DNA bases can alter genes. An altered gene in a sex cell, product of meiosis, can be passed on to every cell that develops from it, causing an altered phenotype, so a change in the physical characteristics. Um, we I showed you those. We don't need to do every single one. We did talk about those. Okay. So your summary of meiosis, you've got your interphase, prophase. Very important part of prophase is the crossing over of prophase 1. Metaphase, anaphase is the breakup. Again, that's also where when the breakup fails to happen properly, you do end up with too many or too few chromosomes. We're going to talk about that in a second. Telophase and then cytokinesis. Now we have two cells with the replicated chromosomes, and then we go through meiosis 2, prophase 1, metaphase 1, anaphase 1, and then we end up with 4 genetically different haploid sex cells. Here's another diagram that shows you the differences between mitosis and meiosis. FYI, this is going to be a short answer question on your exam. Now, let's think about some things that can occur if we had any errors along the way. And typically, that occurs in anaphase when we fail to have a clean breakup. So, genetic disorders are diseases that can be passed from parents to offspring, either because the parent is a carrier, like you carry this in your genes, it's something that is prevalent on your family tree. You could be a carrier who pass the gene for the disorder onto their offspring, or an error can occur during gamete formation. So it's either A, something that you carry on your genes, or B, for one reason or another, an error occurred during usually anaphase. Sometimes entire chromosomes can be added or deleted, resulting in a genetic disorder. An example is trisomy 21 or Down syndrome. We talk about this one because it's one that we have a familiarity with. Chromosomal errors occur because of non-disjunction. It's the failure of chromosomes to separate properly in meiosis during anaphase. Gametes with two extra or Gametes with extra or too few chromosomes result, and diseases such as Down syndrome can occur. So, looking at this karyotype, we know that a karyotype is an arranged pictures of chromosomes at their most condensed state. These chromosomes were collected during metaphase. And we know that this is a male because XY, you're a guy. These chromosomes come in homologous pairs, so one from each mom and one from mom and one from dad. And you can see that there are 23 pairs altogether, 23 pairs, which means there are 46 chromosomes on this karyotype. Okay, so this is a normal male karyotype. Looking at the next one here, this is a image with 23 pairs. There's your XY. So this is also a male. But what you noticed as well is that there are 47 chromosomes here because that the 21st location, there is a third chromosome. So that's where the name trisomy 21 comes from. Tri meaning three, somy meaning chromosomy, and 21 meaning the 21st location on the karyotype. So if you saw this, you would know that it was from an individual who had Down syndrome. Some other genetic disorders that we might see, one is called Edwards syndrome, and feel free to do your own research on these. I'm not going to get into it too much, but Edwards syndrome, you can see, is an extra chromosome on number 18. And again, just picture anaphase. The breakup is supposed to put an even number of chromosomes on both sides. When the breakup fails to happen properly, when non-disjunction occurs, you do have chromosomal disorders. Klinefelter syndrome is when there is an extra X chromosome at the 23rd location. And Patau syndrome, we talked about briefly in Unit 5, that puts an extra chromosome on number 13. Now, not all areas of non-disjunction result in a viable offspring. Sometimes, unfortunately, this does result in the pregnancy not maintaining. So sometimes when the non-disjunction occurs in anaphase, depending on what type of situation we're looking at, sometimes the offspring just doesn't maintain a viable pregnancy. All right. So another way, in addition to non-disjunction, in addition to the disorder happening during gamete formation, another way that a genetic disorder can occur is when it's passed from parents to offspring because the parents are carriers. So an example of a genetic disorder that's passed on by a carrier is Tay-Sachs disease. It's a hereditary disease in which an enzyme deficiency leads to the accumulation of fat in the brain and nerve tissue. It leads to mental disabilities, convulsion, blindness, and ultimately death. And that's so sad. So, what you have here, and we're going to be looking at genotypes. Genotypes. So, the unaffected father, he has a big R, little r, and his little r is recessive. So, he is a carrier for Tay Sachs disease. Unaffected mother, big R, little r. So, she's a carrier for Tay Sachs disease. Sometimes we call carriers heterozygous because she's big R, little r. So, she's heterozygous. And sometimes these are called recessive diseases because they only present themselves if you have both of the recessive alleles. So they don't know by, looking at each other that they're both carriers for Tay-Sachs disease. They would need to have a very detailed pedigree, which is a family tree that shows the passing of genetic disorders from one generation to the other. Oftentimes, when you have a disorder like this in your family, you do go the extra mile to get some genetic information about your family done to see how likely you are to pass that on. Especially if you and your spouse both have family members with the genetic disorder, you might, inquire to see how likely you are to have that passed on or not. It is another one of those ethical, moral, concerns that people are on a spectrum on where they feel like that's appropriate. So it is one of those things. But anyway, back to the genotype. Big R, little r, dad's a carrier. Big R, little r, mom's a carrier. They don't have Tay Sachs disease. They're perfectly fine. When they cross, they have a 20, we could just do a Punnett square here and picture that. There's a 25% chance that one of their offspring could be big R, not a carrier at all. 50% chance that they have a carrier offspring, and a 25% chance that they do have a child who is afflicted with Tay-Sachs disease, which would be very unfortunate. Again, like we saw with the Amoeba sisters, this is how these are predictions. So it is likely that if they had 4 kids, 4 of them have Tay-Sachs disease, just like the Amoeba sisters said, you could have a 50-50 chance of having a male or female, but we all know families where they're like, there's 5 brothers at the house, or there were 5 sisters at the house. So it's always a 50-50 chance with, yeah, boy or girl. But with these types of disorders, there's always a 25% chance with each cross that the offspring could have a disorder. So that's what it means to be a carrier. So those are the 2 ways genetic diseases can be passed on. 1, because of non-disjunction during anaphase, and 2, because you are a carrier for a genetic disease. When you do know you're a carrier, one of the things that can be looked at is your pedigree, which is another way of saying your family tree. And it shows how a disease might have been passed through your family generation. This is hard sometimes because with some of these diseases, it's like the people were mistreated, before, and so with some of these diseases there was like some like people, things we just don't talk about in this family, so sometimes it's hard to put together a family tree because back in the day these types of things were not talked about as openly as they are now. But it is still helpful, especially if you and your partner both have people in your families with a genetic disorder. Putting together a family tree does help you see what your chances are of passing those on. So there's that. And again, as we always say, when we're studying genetics and we're looking at these things, there is the potential for identifying, things that you can't see with your eyes. You're using science and stuff. So you do have to think about like where you are on the moral and ethical spectrum in terms of the application of this tool. So just food for thought, something to think about. All right. I hope that brings us to the end of the lesson part for Unit 6. What you want to make sure that you do is go ahead and actually, I didn't put my slide in for it. I'll do it right now. Let me go get that slide. You guys are I'm not going to have you watch an Amoeba Sisters, if you can believe that, but you're going to work on the next reading and the next lesson for this unit. So you're going to go ahead and paste. You're going to complete the chapter 12.4 reading and the 12.4 reading quiz. Okay, you'll also have some study islands to do tomorrow. You can get a head start on them. And yeah, that's going to do it for Unit 6. I hope you got a lot out of this. I'll talk to you soon. Bye-bye.